We're going to talk about the roles of IL-18 produced by macrophages. First, this is the structure of IL-18. And IL-18 is a member of the IL-1 family of cytokines. And it is similar to IL-1 beta in that it is synthesized as an inactive precursor as pro-IL-18, and it needs caspase 1 to cleave it in order to be active. But unlike IL-1 beta, the IL-18 precursor is constitutively present in nearly all cells of the body in healthy animals and humans. Now, IL-18 has other functions, many functions, which are either in presence of IL-12 or in absence of IL-12. First, we're going to talk about the functions in the presence of IL-12. In the presence of IL-12, IL-18 stimulates the T helper 1 cells, natural killer cells, and NKT cells, which have IL-18 receptors, to produce interferon gamma, which will help in the host defense against microbes, and of course cause tissue inflammation. And there are also two other functions that are not present in the figure, which are that also in the presence of IL-12, IL-18 can act on CD8 T cells to induce them to produce also interferon gamma and lead to their direct activation. And it can also boost the natural killer cells activity, also in the presence of interleukin 12.
Okay.
Now, in the absence of interleukin 12 and in the presence of interleukin 2, IL-18 induces the T helper 1 cells, NK cells, and NKT cells to produce IL-13 and interferon gamma, which will play a big role in the host defense against microbes also and in the infection-associated allergy. And it can also induce these three cells to produce IL-3 and IL-9, which will induce mucosal mastocytosis, which means an increase in number of mast cells, mainly in the mucosa of the intestine. And this will help in cleaning the intestines from nematodes.
Okay.
And now there are other functions that Muhammad is going to talk about.
Okay.
Okay, so to continue about these functions, also NK T cells in presence of IL-2 can act on beta cells to induce them to produce IgE, which aids in immunity against parasites. This action is mediated by the expression of CD40 ligands and the interleukin 4. Also, another role, I don't know, هلا انا الباوربوينت مش مبينه عندي بس انه عم بقولهم anyway. Another role, it also acts on NK cells to increase their cytotoxic activity against the tumor cells. يعني interleukin 18 with interleukin 2 can give a presenting cell-like aspect. And this is done by the expression of certain surface markers like CD80, CD86, and HLA, DR, and DQ. This enhances the cytotoxic activity against the tumor cells. Now there's also an action on neutrophils actually, and according to paper which we read in Journal of Immunology, interleukin 18 receptor is constitutively found on neutrophils. This aids in cytokine and chemokine release as well as the granules release. Now, the last function we want to talk about is inducing an innate allergic response. That an allergic response usually requires an IgE with an antigen complex. And this acts on mast cells and basophils to release interleukins like interleukins 4, 13, and histamine. Now, interleukin 18 with interleukin 33, they can act on mast cells and basophils that have these receptors lead to the release of the same interleukins, like interleukin 4/13 and histamine. And we all know that histamine is involved in the allergic response. That's why we say interleukin 18 have an induced innate allergic response. So these were the functions we want to discuss. So first can show you the references if you want.
Okay, thank you, Muhammad and Faris. So
You're welcome.
you can please send me your the small written paragraph of your answer, okay, so that I can grade.
Yes.
And so we have seen that you mentioned IL-18, one of the most important role is on NK cells, basically to enhance their tumor cytotoxicity, but also other than its role on immune cells, which you have described, there is a new role of IL-18 on goblet cells in the mucosal epithelium.
But isn't that the
sorry?
Neurons? IL-18 effect on goblet cells to increase AMP production, isn't that by IL-18 produced by the enteric nervous system in neurons, which by the macrophage?
Exactly, by the enteric nervous system. Into your assignment was the macrophage.
Yes, by the macrophages.
Immune cell-derived IL-18. True, but just I want to remind your colleagues about the more broad roles of IL-18
Okay.
immunity. So it can act on both immune and non-immune cells. Okay. Any questions from the participants? Okay, so if there is no question, then please stop sharing so that I can start sharing my screen. Okay, so today we'll start with the pattern recognition receptors, which are basically the ones which are involved in triggering inflammation. So everything starts here, basically. If microbes are not sensed by PRRs, then there is no immune response. Okay? They basically pass undetected. And if you remember, when we discussed the difference between pattern recognition receptors and receptors of the adaptive immune system, in the B cell with T cell receptors, we said that they differ in many aspects. For example, the pattern recognition receptors of the innate immune response are inherited in the genome as genes. Okay, so there are independent genes, while the adaptive immune system, the B cell and T cell receptors, are inherited as exons. Okay, there is no genes. Exons need to be shuffled to produce a coding sequence, basically, for a receptor. So we said that they are expressed by all cells of a particular type. Macrophages, almost all macrophages have toll-like receptor 4, for instance. So these pattern recognition receptors can be broadly expressed on cells of the same type, unlike B cells and T cells, which are clonal. So each B cell has a unique B cell receptor. Each T cell has a unique T cell receptor. They trigger an immediate response. Yes, the PRRs are there to trigger an immediate response, while the adaptive system receptors, they need time basically to function. They can recognize a broad class of pathogens, and this is true for the PRRs. And an example, masalan, if we take TLR4, which recognize lipopolysaccharide, it means TLR4 can sense if there is E. coli, Klebsiella, Salmonella, Shigella. So it can recognize a broad class of pathogens. Okay? And this is not true for the B cell and T cell receptors, which are unique for a certain peptide or antigen. They can interact with a wide range of molecular structures of a given type, that's true. For instance, again, if we take TLR4, it can bind to the lipopolysaccharide of many gram-negative bacteria. Okay? Even though they will differ a little bit in their sugar, they can differ not a little bit, significantly in their outer sugars that makes up the LPS. Still, TLR4 will recognize all these LPS, while the B cell and T cell receptors are specific to a certain antigen. So if there is an amino acid change, they cannot bind anymore. Okay, so they don't recognize broad structures. They are encoded in multiple gene segments, no. So PRRs are inherited as genes, while it is true for the B cell and T cell receptors. They require gene rearrangement. This is true only for the B cell and T cell receptor. Clonal distribution, again, the B cell and T cell receptor. They can discriminate between closely related molecular structures. This is true for the B cell, T cell receptor, but not for PRR. For example, toll-like receptor 4 cannot distinguish between the lipopolysaccharide of E. coli and Salmonella and Klebsiella. It will see them as one. Okay, so are these features clear?
Yes.
Okay. So the mammalian, the first class we'll discuss are the toll-like receptors. Okay. Why do we call them toll-like? Because the first toll was discovered in Drosophila, and later the other tolls that appeared, they were called toll-like. And the first indication of a role of toll in immunity came basically from studies in Drosophila. And now we know that tolls are found in many insects, plants, and vertebrates, and they are evolutionarily conserved pattern recognition receptors. So in mice, there are 12 TLRs. In humans, there are 10 TLRs. And with these TLRs, basically, we can recognize a broad class of microbes. With only 10 toll-like receptors, we can sense the presence of a wide spectrum, almost all microbes, if we want to. Almost all, there are exceptions. So what do these TLRs bind? Okay, so if we look at them, if you look at TLR1, 2, so TLR2, first of all, they are all dimers. Okay? All TLRs work as dimeric molecules. Only 2 of them are heterodimers. Okay? So TLR1, 2, TLR2, 6, these are the only 2 heterodimers that exist. Okay? Now, if you look at their ligands, okay, you don't have to worry about the cellular distribution because okay, they are found on innate immune cells, mainly phagocytes, but the table is not complete anyway here. So if you look at the ligands, for example, TLR1-2 and TLR2-6 would share certain ligand. We mentioned at the beginning of the course that pattern recognition receptors, they bind pathogen-associated molecular patterns, so the patterns that are signatures of microbes. Okay? So if you look at the ligands here, lipomannan, okay, so lipomannans, these are complex sugars and lipid produced by mycobacteria. It's a signature of mycobacteria. Okay? And they have a picture here for you. This is a lipomannan or lipoarabinomannan, depending if it has arabinose or not. In general, it has arabinose residues. So you see there is a glycerol backbone here, and then we have an inositol residue. We have 2 R groups again, 2 fatty acid groups, a mannose chain, then an arabinose sugar, and branched arabinose, and on top there are mannose. So it is decorated with mannose residue. So we call it lipo because it has fatty acid chains, lipo, arabino, mannan. So it has arabinose and mannose. So it's a very complex sugar, basically, lipo sugar that is found on tuberculosis. And this is detected by our TLRs in case there is infection with mycobacteria. And of course, these receptors evolved because mycobacteria are parasites of the humans. We carry them basically, so they have exerted some pressure on the immune system to evolve such receptors. Lipoproteins, diacyl or triacyl lipoproteins. Again, these are proteins specifically found in bacteria. And here you have see example. This is a lipoprotein. It's a protein which has 2 lipid tails associated with a cysteine residue, which anchor it to the membrane. Usually they are found in gram-positive and gram-negative bacteria. They can be anchored in the cytoplasmic membrane or the lipopolysaccharide membrane. Okay, so they can be anchored in both, and they play diverse roles in bacterial physiology, adhesion, transport, and so on. Okay, so they have several roles, and when bacteria divide, they do shed some of these. So toll-like receptors do not bind these molecules when they are inserted in the membrane, because as you see, the toll-like receptor, toll-2,6 and 2,1 dimers, they need to bind the free molecule. Okay? They need to interact with these fatty acid chains. So TLR2,6 dimer binds the diacyl peptides, so with the 2 fatty acid chain. TLR2,1, they bind the triacyl, and you see here the 3rd fatty acid chain basically cross-link the 2 molecules together. So again, these peptides are a signature of gram-positive and gram-negative bacteria, and they are sensed by these two molecules, 2,6 and 2,1. TLR3, it senses double-stranded RNA from viruses. Okay, so a double-stranded RNA genome, and these long double-stranded RNAs, they are usually absent from our cells. They are found in viral genomes. Toll-like receptor 4, it binds lipopolysaccharide from gram-negative bacteria. Again, this is a signature of bacteria, but also recently lipoteichoic acid from gram-positive bacteria. So these are decorating units or polyalcoholic, polyhydroxylic molecules found on the surface of gram-positive bacteria. TLR5 binds flagellin. This is the protein that makes up the flagella of bacteria. Again, this is something unique to bacteria. TLR7 binds single-stranded RNA from viruses. 8 also single-stranded RNA from viruses. And TLR9 binds to DNA, okay, with unmethylated CpG. So what do we mean CpG? So a DNA which has CG motifs that are repeated, CG, CG, so on. Okay. And some of this DNA is found in DNA viruses like herpes viruses, cytomegaloviruses, and so on. TLR10, we don't know what it binds, and TLR11 is only found in mouse. You don't have to worry about it. It binds specific proteins from parasites that usually infect mice. So you see that this is what we call PAMPs. Okay, these molecules that are found here, they are PAMPs. And if you look at the PAMPs, do you see any indication of a toxin? Is there any bacterial toxin among these PAMPs? Are there any toxins? There are no toxins. Okay, so these PAMPs are basically molecules, sugars or lipids or nucleic acids that are specific to microbes, okay, that are a signature of the microbes. Okay. Any questions on these?
Professor?
Yes.
Why would we have two toll-like receptors that have function, like TLR7 and TLR8?
Why do
what
we
would
have?
be the use?
But why 7 and 8? It's a good question. We don't know yet because they do bind similar. They are activated in response to certain viruses. So certain viruses can activate both of them, 7 and 8. It's not clear. We have no answer for it. It's a good question. But again, their distribution, if you look at their distribution, is they are not distributed on the same cells. How much is the redundancy? So they are not completely redundant. So again, remember, these are, now when we talk about the structure of the receptors, you can understand better. So they might recognize certain RNAs better than others. Okay, so although they can recognize same viruses, but we don't know exactly what viruses are recognized by 7, not 8, and vice versa. But they should exist, otherwise these genes will not have evolved. We'll talk about it now in a second, okay?
All right.
Now, if we look at the compartmentalization of these receptors, and this is important, where do we find these toll-like receptors, okay? They are always membrane bound, okay? So they are either found on the cytoplasmic membrane on the surface of the cell, or they are found in the endosomal membrane, okay? Now, if you look at what TLRs are found on the surface and what TLRs are found in the endosome, you see that there is a clear compartmentalization. So TLRs that recognize bacteria and fungi are found on the surface of the cells. TLR4, 5, 21, 26. Okay, if we go back to the table, 26 can recognize zymosan, which is a complex glucose chain found on fungi, fungal cell walls, but also glucan chains, other glucan chains found in fungal cell walls and bacterial capsules. So TLRs which recognize bacteria and fungi are on the surface of the cell. TLRs which bind nucleic acids are endosomal. Okay, so nucleic acid TLRs 3, 7, 8, and 9, which 3, 7, 8 recognize RNA and 9 recognize DNA. Okay, they are found in the endosomal compartment. Why do you think these nucleic acid TLRs are all found in the endosomal compartment? They are not on the surface of the cell.
because viruses replicate بقلب السيل.
Viruses replicate بقلب السيل.
Viruses inject their genetic material in the cell.
We'll come to that later. We'll come to that, how the viral RNA comes in contact with these TLR. But what's the important reason for this compartmentalization? TLRs that bind bacteria and fungi are exposed to the outside of the cell. They are on the surface, while nucleic acid binding TLRs are found in the endosome. They are not exposed to the outside.
They are acidic. المفروض يكونوا باندوزوم.
لا, what would be the danger? Remember, let's take the question from you. If you want to recognize a virus, okay, so the virus is not like a bacteria. It doesn't have specific sugars like lipopolysaccharide, teichoic acid, peptidoglycan. What is a virus? It's made up of protein and nucleic acid. Okay, and if the virus is enveloped, the envelope comes from the cell, from the host. So the lipids are similar to those of the host. So can I recognize the viral proteins? We have thousands of these proteins, and we know that proteins are not ligands for PRRs. A protein is a protein. Okay, we eat lots of foreign proteins. We are exposed to lots of foreign proteins. They don't activate the immune system. So if I want to recognize the virus, okay, the only structure of the virus I should be able to sense is a nucleic acid or the genome of the virus. Okay. But what is the danger in such a strategy? Only viruses have RNA.
No.
So if I designed a receptor that can bind single-stranded RNA, what would be the danger?
دكتور بس انه مش this receptor انه ما it's to the outside to
لحظة, they are inside, they are in the endosome. The nucleic acid receptor in the endosome. But what is the danger if I evolved a receptor? The immune system decide to create a receptor that can sense a single-stranded RNA from virus.
For the messenger RNA.
Exactly. How does the single-stranded RNA differ from the messenger RNA, tRNA, ribosomal RNA in the cell? Isn't there a danger of cross-interaction?
بس مش the receptors are going to be to the outside?
When outside? They are in the endosome, هون جوا. They are not exposed to
the scenario that
I mean, why do you think why they are in the endocytic compartment? Why these nucleic acid receptors? So that you understand racial compartmentalization. Why they are compartmentalized? So that they are.
الviruses come inside as a compartment. يعني إذا جبناها
Exactly. صح. The viruses will get in the endosome, but to put them away from the messenger RNA, ribosomal RNA, tRNA, which are found in the cytoplasm. Okay, our single-stranded RNAs are found here, and this is the reason why these nucleic acid TLRs, they are not soluble, they are not in the cytoplasm, they are found in endosome, and this is the recognition head. It is in the endosome. This is the signaling part. Okay, so the fact that they are packed in endosome, first of all, is to put them away from contacting our RNA material. First. Second, the fact that they are not on the surface. What happens if these were on the surface and in the vicinity of this cell, an apoptotic cell ruptures?
كمان they will bind the self.
Of course, having them on the surface, not all of our apoptotic cells are removed. Sometimes some they become necroptotic because there was a delay in the removal and so on. So if these nucleic acid TLRs were on the surface, the probability of them contacting self-DNA and self-RNA would become higher. And this is not good because this can lead to autoinflammation, but also to autoimmune diseases. And now we know that TLR9 is involved in several autoimmune diseases, in initiating autoimmune diseases. So this is why the immune system evolved receptors to bind viral RNA and DNA, but at the same time has placed these receptors in the endosome so that they are not in contact with the DNA of the host and the RNA of the host as much as possible. Okay, and this is the logic behind their compartmentalization. Now, how do viruses reach these? Where do viruses encode? Again, if I ask you a question, how does the viral genome gets in contact with these TLRs? But the viral genome is hidden inside the protein capsid. Who can explain a possible scenario?
Maybe when the virus replicates its nucleic acids وبعدين it spreads them يصير في كل nucleic acid forms its own protein.
Yeah, but keep in mind, when does the virus reveal its genome? بالendosome أو بالcytoplasm?
In the cytoplasm.
In the cytoplasm, viruses uncoat. We call it uncoating. Yani they remove the protein coat so that the genome can be translated, transcribed, and so on in the cytoplasm.
Can it be due to the acidity? The envelope is degraded, so the viral RNA is released.
Exactly. So what is happening here in the endosome is that when the viruses are endocytosed by a macrophage or a dendritic cell, so they bind to a receptor, they are endocytosed, or they are brought in by macropinocytosis. What happens is that some viruses might escape from the endosome, try to replicate in the cytoplasm, but some viruses will eventually move to more acidic endosomes where the acidity will disrupt their protein coat, revealing their genome, and these endosomes fuse with endosomes containing TLRs. So now the TLRs come in contact with the viral genome and start the signaling process in the cytoplasm. Okay, so viruses eventually that are trapped inside endocytic vesicles which acidify start to disintegrate their code, eventually release their genomes to be sensed by TLRs. Okay?
Professor,
Yes.
does that mean that it can't recognize like the toll-like receptors, the nucleic acid ones? They can't recognize the viral nucleic acid inside the cytoplasm?
لا لأنه هن منو بالcytoplasm. These receptors, you see their head is in the endosome. It is not to the outside. It's not to the cytoplasm. So the genomes in the cytoplasm are not recognized by the toll-like receptor. So now what happens if viruses, if you have infected with a virus that escapes early on before acidity starts into the cytoplasm, will it be detected by the TLRs?
No.
No. So this means, do you think that TLRs are the only viral receptors we have in our cell, or we should have other receptors?
لازم يكون في others.
There should be others. Okay, and this is what basically scientists found, that while studying the TLRs, because this was the first group of receptors that people had their hands on, they found that when they mutated certain TLRs, viral TLRs, they found that the response to certain viruses was intact. It means that there was another receptor responsible for that, and then the search for cytoplasmic receptors, because viruses are found in the cytoplasm, started, and we'll talk about these receptors later after the TLR. Now let's look at the structure of the TLRs. An important thing to keep in mind is this horseshoe-like structure. They are known as the horseshoe-like receptor, and this is made up of leucine-rich repeats. Okay, it's a very important domain made up of 18 to 25 leucine-rich repeats. So each rectangle here, each square or rectangle is a repeat, which is rich in the amino acid leucine. So leucine-rich repeat proteins or domains are highly versatile. Okay, so so we might disconnect in a minute. Please connect for the second lecture. Okay? So these structures here, leucine-rich repeat domains, are highly versatile. What do you mean in biochemistry, highly versatile? And they can bind to many different types of ligands, ranging from sugars to nucleic acids to liposugars, etc. Okay? So you see that they have many repeats. And for example, if you go back to the table, and I tell you that TLR4 binds lipopolysaccharide and lipoteichoic acid, you will tell me how, because these two structures are completely different. Teichoic acids are polyhydroxylated compounds, while lipopolysaccharide is a lipid, has fatty acid chain and a long sugar and sugars. How do these structurally different molecules bind to the same receptor. So if we look at the TLR4, okay, you see it has many TLRs, many leucine-rich repeats. So it could be that, let's say, repeat 3 to 9 is dedicated to bind LPS, while repeats, let's say, 9 to 12 are dedicated to bind teichoic acid. So it can, such a receptor can dedicate different repeats to bind different ligands. And this is how these receptors, this is how a single receptor can interact with different molecules. Okay? It is unique to the this leucine-rich repeat receptor. Is it clear, this point?
Yes.
Okay, so this is something I want you to keep in mind when we talk about TLRs, is that the type of domain they have, the leucine-rich repeat domain, which allow them to dedicate certain repeats to bind a certain ligand,